Amy saw you through.
A little cranky.
Well, welcome to day 2 of the International Space Medicine Summit.
It's early on a Saturday.
The first panel today is Genomics and Spaceflight. I've been looking forward to this panel for a long time. I'll let Mike give you the reasons why it's important, but I really think in medicine that we're at a fundamental shift now in how we do prevention and screening and treatment and medication choices and dosage, and I think it'll also impact who you pick to go on a long duration spaceflight where there's limited ability for care. We're just at the start of it, but I think it's going to be dramatic. Mike Barrett is, of course, a longtime friend and colleague, and he's the kind of person that if you need something done, you go to, and whether it's HRP or establishing medicine in Russia for the United States space program or flying in space, you go to someone who gets things done, and Mike gets things done. For those of you from the United States will be very familiar with this illustration, but Mike's really a good example of the illustration of the father that had 2 twin sons. One was the optimist and the other the pessimist. And the father gave the pessimist a new horse for Christmas, and he gave the optimist a pile of horse manure. And he came back 15 minutes later, and the son that was the pessimist with the horse was crying, and he said, "Why are you crying?" He says, "Well, I'm going to have to feed the horse, I'll ride it, and I'll fall off and I'll get hurt. It's going to have to go to the vet, and then eventually it's going to die." And he went to the other room where the optimist was, and this son had a shovel out and was flailing away, digging through the manure, and he was singing and happy. And he says, "Well, why are you so happy?" And he says, "With this much manure, there's bound to be a horse in here somewhere." And Mike is that son. That the reason he gets things done so much is he's so optimistic and so eager to do things. And I'm hoping that the space program will give him a chance to actually go back into space and find out if the IP of VIIP actually is a factor or not. So I hope there's more for him to do yet.
Thanks, Richard. So I think when you think of me, you think of horse manure, so that's kind of recalibrated everything. What I didn't tell people about Richard yesterday is that he had, 3,290-ish hours of flight time, which is pretty respectable, not really high, but he soloed at 4 hours, which is pretty amazing, or pretty dangerous. So anyway, so I think we got our panel assembled, and I agree with Richard. I think we are kind of at a crossroads in medicine and certainly in space medicine, and this whole rubric of genomics has really taken medicine by storm, but not in a necessarily controlled or understandable fashion by all of us. Now, most of us know that the human body undergoes this grand constellation of changes when it gets into weightlessness. And we've got a pretty good handle on most of these changes from a gross level, but they come in stages and different flavors. So some of them, like neurosensory changes, are just adaptive. They don't really harm the body. They do become maladaptive when you want to come home, and it messes you up for a little while, but fairly reversible. And some are a little bit worse for you, bone and muscle loss, that we have to counter. We have to actively provide countermeasures for, and then we know when you come down, you're going to have a rehab time period to try to get back to a pre-flight baseline of health. And now there's others, and we know more about these now, that are a little bit more sinister, like the vision intracranial pressure issue, or SANS, or MOC, or SOS, or whatever you want to call it nowadays. And these are things that actually can cause dysfunction or degrade performance when you're on board. So some of these we do counter, and we know that grossly it's an issue of removal of physical load, and we can grossly put back physical load, and so with that we can prevent some bone and muscle loss, but we can't kid ourselves that we understand mechanistically what's going on. So we're really taking a blunt instrument approach. We don't know why bone resorption and bone formation are uncoupled. It's really quite amazing that they are. But we do know that if you provide those loads, you can actually maintain bone and muscle pretty well. But developing a complete mechanistic understanding of these changes has eluded us somewhat. And in doing so, we could actually become more on the offensive rather than the defensive, and hopefully move us towards more of a predictive and away from a reactive mode in dealing with some of these changes. Now, I'm talking about weightlessness, but there's other things that also induce mechanisms which compromise human performance. These could be radiation, for instance, noise, isolation, stress, environmental gas constituents. And so we have a broad need for complete mechanistic understanding of physiologic responses that is kind of unfulfilled at this point. So genomic analysis really offers us a powerful tool. It's a technique that I think most people know has matured tremendously over the last decade and a half or so. And being able to map gene expression to what we know physiologically and biochemically, again, offers us the chance to gain this mechanistic understanding. So what we're looking for is mapping our changes to the genetic expression and all of the downstream changes. Now, often we flip the term omics around pretty loosely, and it's a vernacular that some understand, some don't. I still get a lot of questions on it, and I'm not a genetics guy. So I actually had to go look it up, the etymology of omics, and it typically means disease or some tumor like oma, as we know, but we use it biologically basically to mean the study of the totality of. So we're talking about the totality of genomics, the totality of proteins, proteomics, et cetera. And if some of you have seen the recent moniker in the internal medicine literature is the diseasome of physical inactivity, which includes diabetes, hypertension, and other things associated with being overweight. So that's kind of how we use it. But in genetics, more and more in the popular press, we're using the buzzword just omics for gene expression, all the resultant products along the chain of the central dogma, genomics, transcriptomics, proteomics, metabolomics, and whatnot. And that's what we apply to our knowledge of the microstructural and biochemical level. What we stand to gain out of genomic studies, especially when you look at spaceflight. We should get an increased general knowledge about the specific questions related to our world, which is microgravity and everything else associated with spaceflight that we mentioned. With that, an insight into the personal health risks, because gene sequencing obviously is a very personal diagnostic tool. Hopefully we will find specific diagnoses of the latent genetic diseases that could lead to susceptibilities to some of these environmental factors, and hopefully to preemptive treatment or intervention. We'd like to apply these to animal models whenever we can, because obviously we have a much greater access to tissue available. And I don't want to sell short the microbiome associated with the individual and with spaceflight and what we can learn from genetic analysis. But I think everyone knows this comes with baggage, like every great tool. So we worry about the generation of information that's highly personal and highly attributable and on a fairly grand scale. So rather than a specific data point like somebody's cholesterol level or biopsy results, if you will, genomic data can be reconstructed into a pretty specific profile that describes an individual, almost every aspect, morphologically, phenotypically, and probably their political leanings if we learn how to do that better. And this has been demonstrated with publicly available genetic information sequenced on individuals, where others were able to go back and actually find the addresses of these people based on reconstructing their physiologic attributes. So we worry, privacy, insurability, anxiety over the implications of the results. You can learn way too much. You could learn a lot more than you're asking, and that adds some complications. So as so often, the powerful tools come with formidable complications, and they require meticulous control and consideration as you use them. So I would say that some of these concerns have kept genomics out of the spaceflight world for several years longer than they should have. And maybe that's appropriate. We've actually been able to pause and build good genomics policies and guidelines to go forward. At the same time, our clock is ticking. Our N is low. Even though we have a high profile, our N is low, and we really want to apply the most powerful techniques we can. But the payback is just so great. The insight, the information that we can get out of this really makes it worthwhile. And again, I would argue that it's past time to apply these techniques to space life sciences. So we have assembled an august panel of people who are actively engaged in certain aspects of genetic research. And I'm going to do the same thing as yesterday. Each one will get about a short introduction, less than 1 minute. Not to belittle your august careers at all, but after that, give us about 5 or 7 minutes or so of thoughts and activities, and then we'll open it up. And again, I think everyone understands this is a very candid forum, and this is a chance, an opportunity to ask questions that I think you'll get very good answers on. Craig Kundrat, I will skip some of the lines that he gave me for his bio, but in his spare time, he's the director of Space Life and Physical Sciences Research and Applications, probably the worst NASA acronym ever, SLPSRA, I guess we can call it. So he's at NASA headquarters working under HEOMD. Craig has a bachelor's degree from Northwestern University. Go Cats, I'm actually an alum of that as well. And a doctorate from Yale, an erstwhile structural biologist who studied protein and RNA structure-function relationships using X-ray crystallography from the University of Colorado. He joined NASA's biotechnology program in 1998 at Marshall Space Flight Center as a senior scientist before assuming science management positions for biotechnology and materials science. He came to us in 2006 to the Human Research Program at NASA Johnson Space Center. He has worn many hats, but officially he was the Deputy Chief Scientist and the first mission scientist for the HRP twin study. He served as chair of the Institutional Review Board at JSC and helped us and guided us in formulating a NASA genetics policy for astronauts. In 2015, he became the life sciences lead in the Office of the Chief Scientist at NASA headquarters and could only handle that for a year before we tapped him to direct SLPSRA. And Craig, along with the other experience you've had, you're now overseeing the Gene Lab project, and I wanted to get a few words on that if we could.
Okay, thank you, Michael, for the opportunity to speak today, and Mr. Abbey, thank you also. So Gene Lab was created several years ago for the reasons Mike just mentioned, to get at mechanistic understanding in biological systems, and also to maximize our use of spaceflight data, because omics is so rich, and just as Mike said, we're talking about the genome, which is pretty stable, but the transcriptome, proteome, metabolome, of various tissues and cell types. And Gene Lab was established to capture this data and provide tools for analysis, and most importantly, capture the metadata that go with a spaceflight experiment. There's a myriad of data. You mentioned many of the environmental factors that are associated with a spaceflight that the typical ground investigation doesn't have. And without knowing what those metadata are, one could mistakenly not take into account confounding variables. So Gene Lab was established. It has in it data from microbes, plants, fruit flies, worms, mice and rats. And it is structured so one day it can accommodate human data as well. However, in the NASA world, for reasons Mike alluded to, there is a huge difference between what we do in the space biology realm with non-human data and what we do with human data. So when it comes to human data right now, that is fenced off, and the twin study is a great example of that. That really helped shape the agency's policies because, as Mike alluded to, we were kind of taking our time getting into the omics realm, but this unique opportunity to study the twins really hastened that along. So as Mike alluded to, when it comes to omics studies or genetic studies in general, the primary risks are psychological and social, not physical. And the astronauts are a special subject population, unlike your normal NIH study, which involves hundreds of people probably, at least, and are basically from all over the place and relatively anonymous. Astronauts are public figures. There are people out there that are basically data stalkers that try and collect as much information about astronauts as they can. And so protecting the astronauts' privacy and the confidentiality of their data is a real challenge. So unlike the Gene Lab world where we have broad intake of data and make that open to the wide world, if you're interested, all you have to do is Google NASA Gene Lab and you can promptly have access to all manner of data. In the human spaceflight world, it's very different. And just to summarize some of the protections that were put in place, they basically fall into 5 categories. The first is a very broad definition of what we mean by genetic research. So it's not just gene sequencing, but also when studies involve study of the transcriptome or proteome or metabolites that one could make genetic inferences from. NASA includes that in its definition of what a genetic policy is. So it's a very broad application. Second, for these studies, they are restricted to what they propose to do in their institutional review board protocol, and nothing beyond that. And that includes, for example, not trying to identify de-identified subjects, which is not really an issue in the twin study because you don't have to be too sharp to realize who's who. But in future and broader studies, trying to de-identify who's who out of a cadre of a dozen is not permitted. Third, when it comes to the actual samples collected or material generated in a study and the data, NASA reserves the right to say, thanks very much for doing the study. We will now take that material and or that data as NASA, and you will destroy any copies of data you have and the rest. So we reserve the right, if it comes to it, to basically sequester samples and data. Because omics and genetic interpretations is a fast-moving area and there's a lot of subtleties involved, we also provide as a fourth aspect of protection, if you will, genetic counseling. So the research subjects know what their data are saying or not saying, what inferences can or cannot be made about parents, siblings, children, et cetera, because that's another dimension to the ethics. It's no longer just the individual who's sharing information, but in some inferential way, you're sharing information about others. And then lastly, the greatest control of all is any publications or presentations involving this genetic information construed quite widely is subject to the review of the subject, who can ask for identifying information or aspects to be deleted from either the presentation or the paper. So our approach basically in 2014 to prepare for the twin study was to take this very conservative, protective approach where we had lots of mechanisms to rein things in and insert gates. And it has served the twin study very well so far. Going forward for larger studies, we may have to take a look at it and see how practical all the elements are, like the degree of genetic counseling that we've been able to provide to the Kellys might not be sustainable in a much larger study, but that's our starting point, is basically to err on the side of protection, and as we get smarter about how to treat data and in this field and how the astronauts as subjects feel about it, we can relax as is appropriate. So that's in stark contrast to GeneLab, where again you'll find all manner of biological organisms and their spaceflight responses, and equipped with lots of metadata to go along with that. So we really have two separate tracks when it comes to omics or genetic type studies. We have the non-human, very open and accessible and complete, and then in the human realm, it's very tightly controlled to the investigators and with a lot of involvement from the subjects on what can or cannot be released.
All right. Thank you, Dr. Kundrat. I'd like to next introduce Dr. Vladimir Saichov. Vladimir Nikolayevich received a PhD from the Moscow State University in biophysics in 1974 and came to the space program. He has very extensive experience in biomedical investigations in flight. We'll let everybody get wired up here. And has been deputy director of the IBMP for biological investigations on board ISS as long as I've known him. He is chief of the Bion-M program, very successful Bion satellite program, and a very exciting prospect with that is a flight in, I believe, 2021 to 1,000 kilometers, which is going to be much higher altitude and exposed to much more radiation than we've had in the past. So an incredibly sharp tool and a program that's been very successful. And I'd like Dr. Saichov to talk a little bit about the Russian investigations into animal studies and genetics.
Thank you. Genetics, to my mind, is one of the main fields
Because if bio program that
came
together in the 70s, it was mostly based on zoology. Right now, we are aiming at genetic research mostly, and we often get questions to which we cannot get answers. If we take mice and animals, we found out that for them genome reaction is quite surprising and amazing. If we believe that the muscle tissue has to do with unloading, with mice we found out that it has to do not only with unloading muscle. There is a cascade of genes that is responsible for it. Even those that do not have anything to do with the muscles give us answers. Also, more amazing results were achieved with research of fish with the Japanese. Request to speak slower because I'm not able to follow. Okay, the research with fish. Although fish has no mechanical application to muscle, we have the same picture here as land-based animals. The idea is that it is not a physical application, but a genome responsibility to see what is happening. The changes that we see in animals makes us think that not only physical and physiological reaction of a human to weightlessness may be present, but also genetic elements are more important. And if so, then we will have to revise perhaps the system of countermeasures. Also, as an example, I want to bring something to see why genetics is so important in the space field. We know that if the expedition is a long duration expedition, then physiological rest may help person to endure the long-term flight. But however, the experience with animals tell us that we have some surprising results because the 1 month exposure of embryonic cells of maxillary pod shows us that they fine at first, but when they return to the ground, the second generation gets male species, which is basically impossible on the ground. It may happen only if the previous generation experienced a lot of stress. So I am still surprised why, if we didn't have any active molecules at rest in spice, it still occurred. I believe it is a genetic responsibility. Also, we have an idea that not only weightlessness and physiological other factors are in play here, but also there is a genetic field that is encompassing all kinds of processes in the organism. Thank you.
Vladimir Nikolayevich. Okay, great. I'd like to introduce Mike Roberts, Dr. Michael Roberts next from CASIS. He is the Deputy Chief Scientist of CASIS, the Center for the Advancement of Science in Space, where he works to imagine and enable science and space for life on Earth, utilizing the ISS as a national lab. Prior to joining CASIS, Michael worked as a microbial ecologist, a molecular biologist, and a group lead and PI in NASA's Advanced Life Support Program at the Kennedy Space Center. He conducted flight experiments on shuttle and ISS, focused on bacterial recombination, plant microbe interactions, and forward osmosis membrane technology for water recovery in microgravity. He has a bachelor's from Maryville College, a PhD in microbiology from Wesleyan University, and was a postdoctoral researcher at the Center for Microbial Ecology at Michigan State University and the Riken Institute in Waukesha, Japan. I probably butchered the pronunciation, but I asked Dr. Roberts to give us some insights into the genetic research, the genomic research that's utilized in a different channel from the Human Research Program and others using the ISS as a national lab.
Thank you, Mike, and I'd like to thank Mike and Mr. Abbey for the opportunity to be here with you today. So CASIS's role, as I highlighted yesterday, is to provide access to a broad user community to utilize the unique environment of low Earth orbit to address problems that are of interest for the benefit of Earth. So our research support not only is slightly different than the focus of NASA on exploration goals, where the focus is on enabling exploration of space, making sure that crew members and tourists and others who are interested in going to space have the ability to perform and enjoy that environment to the best of their ability. So with our focus audience being very broad, we've obviously worked very hard to enable access to the International Space Station for a variety of research efforts. And foremost among those is the interest in genomics. As Mike alluded to in his introduction, genomics is essentially best thought of from my perspective as all the genes all the time, all the proteins all the time, all the metabolites all the time. So in a grand scale, the goal of all that study is to understand and develop correlations between what's going on at a genetic level with what's going on at a phenotypic level. So we now enjoy the ability with a variety of platforms, a variety of models, a variety of tools, some of which are actually available for use on the International Space Station, others which are available only on the ground, to design experiments where the focus of the inquiry is directly on what's going on at a structural level with DNA, what's going on at a transcriptome level in the DNA and RNA, what's happening with the proteins that are generated from those either dynamics in gene expression, as well as moving into the foray of metabolomics, where you're able to look at small and large molecules that have impact upon the phenotype of the organism in that environment. As Craig mentioned, there are a variety of efforts, perhaps culminating most recently in the data set that's generated from the twin study in 1 year in space, that make excellent use of samples that were generated by the crew. And the ISS National Lab can also provide access to those. But those samples are precious, they're limited. I think we're all still struggling through issues related with genetic consenting that Mike had mentioned. There are still questions about how best to enable access to those samples and specimens going out, because they're in such high demand, and we're living in an era where we're still trying to define what the appropriate standard measures are for using those systems. So given the limitations in accessing those human samples, what we've seen at CASIS for the ISS National Lab is an interest from other government agencies, such as the National Institutes of Health and National Science Foundation, as well as commercial sector companies, primarily among those large pharmaceutical companies, in utilizing model systems that Dr. Kundrat mentioned as well, and my colleague to the left mentioned. So there's been ample opportunity and interest in utilizing model systems. First and foremost, and at least from the phylogenetic perspective closest to humans, are the use of rodent models in the space environment, the use of flatworm models for regenerative medicine, the use of Drosophila-based models for inquiry into cardiac function in the microgravity environment, to holistic approaches where try to look at microbiome from a population perspective and understanding the interaction between the organisms that live within us and on us and in our environment, and how they impact the health of all of the individuals in that system. So we have a variety of engagements with pharmaceutical companies utilizing the rodent model, and every single one of those investigations not only are interested in looking at the physiological effects and phenotypic effects of the spaceflight environment on that model organism on the mouse, typically. But they're also taking genomic-based approaches to understand at a systems biology level, at a systems level approach, how those changes in spaceflight directly impact organism health and disease status. So one of the significant advantages of using those rodent models and others is that even a few weeks exposure, a couple of months of exposure to the spaceflight environment represents several years in the life of those mice. You also have the ability of thinking about them essentially as the twin study to the power of infinity, because you essentially have organisms which have very high genetic similarity. I wouldn't say identity, but they have a significant share of genetic similarity at the start of the experiment. So you can track changes in transcriptome level or the level of epigenetics that manifest themselves very quickly in those populations. So we at CASIS are seeing significant interest in genomics approaches. Everyone that is utilizing those different model systems is understanding the correlation between the measures that they obtain and the phenotype, really understanding how those impact the health of the organism, and then extrapolating those to humans. Because at the end of the day, that's where most of the interest is in understanding what in the spaceflight environment is causing those changes and how they directly apply to human health. Thank you.
All right, thank you very much. It's very interesting. Next, Dr. Jennifer Fogarty. She was introduced yesterday, so I'll just keep this very brief. I'll just mention she's earned her PhD in biomedical sciences from Texas A&M. I met Dr. Fogarty when she was, I think, fairly new still at JSC, and she spent about 10 years as a translational scientist trying to bridge the gap between medical research and medical operations, which may come as a shock to you, but those two entities don't speak with one another as well as they should. And Jen did a great job of trying to integrate information between those two parties. We rewarded her or punished her as you see fit by making her the chief scientist of the Human Research Program. And she's new in that role, but we have great expectations for that. And I've asked Dr. Fogarty to give a few words about the relevance of genetics, genomics for human research insight.
Thank you, Dr. Barrett. Thank you, Mr. Abbey, for inviting me to participate in this event. I have to thank my prior speakers because I think they did a really good job at describing the breadth of the landscape about that word omics. And so HRP has, a very applied charter. And you heard part of it yesterday talking about countermeasures. We do try to understand how to contribute to preventive measures. We have responsibilities for understanding long-term health implications and how you would manage those as well. Under a restrained and confined budget, the portfolio that you invest in is really challenging at times for how to spread yourself so thin given the breadth of what we were talking about. So we really rely on partnerships. Dr. Condrey from SLPSRA oversees space biology as well as physical sciences, and with HRP in that domain. So we really look to space biology as one of our partners when you talk about non-human models of understanding the omics presentation and targeted genetics potentially of what we could look at in a more refined way in the human cases also, wherever the evidence is going to come from. But we heavily lean on terrestrial academic medicine to make that connection between identifying changes in gene expression, ultimately leading to protein of interest that will affect the phenotype, the outcome, and understanding the uncertainty, which is quite large at times. I always have to do a caveat because, to remind folks, because I think the pressure of the mass media nowadays and kind of the headlines they put out and what they choose to highlight in different studies that come out is genes are merely your potential. They're not necessarily your destiny. And we look for expertise to help inform when those things cross that threshold to actually being deterministic, which are very few. Of the 50,000 plus genes that we have as humans, only 55 are truly actionable at this point. That means you have to have the evidence base and the certainty that if you're going to act on someone's gene profile, you're pretty sure 99%, greater than 95%, you understand the effect you're going to have to change that outcome. Now, I think we are all comfortable dealing at the phenotypic level where you see subclinical changes or risk factors. In my background, coming from cardiovascular disease, I always go back to the model of the Framingham risk score model. And you know those things with a high degree of certainty affect outcomes such as heart attack or stroke. So we know not to smoke. You can't do anything about your age. You can't do anything about your the sex that you were born with. But there are other factors. You can modulate your cholesterol. You can do things to reduce inflammatory processes in your life. But clearly there's genetic underpinnings in there that for all of the dials those things represent, you can turn some and you can't turn others in any one person. I think we're trying to get to the point of refining for space flight, what are the dials, which ones can we turn and which ones we can't, and which ones aren't we going to be able to turn for an individual, but that doesn't mean they don't fly. So yesterday, I think Dr. Ginges brought up the topic quite nicely about you could identify a gene and realize that it's a different risk for that individual, but we may not be able to accommodate it in flight, let's say. If someone absolutely needs a treadmill and exploration isn't going to have a treadmill, what are we going to do about that? Well, then we rely on other countermeasures. This is where potentially pharmaceuticals come into play, other types of stimulus for that individual. So we can refine it and change some of those dials. But I think it also goes back to a lot of the conversation we heard yesterday about risk balancing and informed consent, helping people know what the probabilities are, again, without saying it's an absolute certainty. So we've also invested in things. HRP was the sponsor for the twin study. So while we've grappled with the conflict and the lack of N number we're going to have, we're not the ones building the database, to be honest. We're getting these little insights potentially, but what they mean will be much harder to tease out in a population of 50, maybe 75 ultimately when we pull all the data together. But terrestrially, you look at the extreme stresses that humans undergo in different paradigms, whether it be Antarctica or climbing Mount Everest, suffering from extreme disease states. And those can be very informative in terms of what do those genes mean with respect to outcome. So I think Mike's comment about correlation, they're interesting, but they're not causality. And again, that's language that sometimes get confused, that you could be measuring a change in a gene expression and an outcome, and they're correlated, but it doesn't actually mean that that gene caused that outcome. It's in the rolling around in the context of it, but it may not be the site of action. It may not be root cause. So when you think about 50,000 genes, you have gene-on-gene interaction, gene-on-environment interaction, which is epigenetic. You have microbiome, which isn't your genes, the genes of the bacteria that live in you. We hear more about the virome, viruses that we're commensal with and do provide a beneficial influence on our constituency, that now we're talking 50,000 to what power? So trying to sort through that can be a little overwhelming. So you step back and say, okay, for our purposes, what are we going to zero in on? And we actually have some examples of that specificity. So Dr. Scott Smith was here yesterday. He has advanced the concept of a polymorphism in a homocysteine regulated gene that we think is of interest, and he's published papers on its relevance to the VIIP or the SANS issue, and he's pursuing that, and that's an area of interest. We have other polymorphisms that are coming out of, say, the space biology world or the medical terrestrial world that then would become a primary point of interest for us to follow up. The twin study was really full spectrum. Let's go map this thing out for the people who can participate. We had one person flying, the other person staying on the ground. How did they differentiate? And those results will be coming out. We've heard different things, when they've pre-released some preliminary data. It is interesting, but it's just it's noisy, right? Where is the signal in all that noise? And probably from the twin study, we're not going to figure out signal per se. So it's an interesting evolution into the world of using genes, but I think we're guardedly optimistic about when we'll be able to act on them. And it's okay if it's not in the next decade. What's important is that we invest in it, try to understand it, and build from there. No one wants to stick their head in the sand, but I have to say, back to Dr. Barrett's comments, this is considered a small vulnerable population. And when you deal with genetics, you actually don't just impact that individual, but then some of their family members. So that's why the data is guarded so vigorously. And at this point, we have a lot of constraints about how we would use that data, who would get to see that data, and it's not in an open place for people to go in and kind of go fishing and see what they would find. And I don't see us going there anytime soon, for sure. But it is, we have acknowledged the need to do this work. We're stepping cautiously forward in it, and we're definitely seeking expertise to help guide us both in the medical and research domain.
Okay, thank you so much. Very insightful. All right, next, Dr. Kate Rubins. Boy, I think I was the first person in space to go from the shuttle middeck to the HTV, which was docked, the ATV, which was docked after the service module in less than 30 seconds without hitting anything. Kate was the first person to sequence DNA in space. So, mine pales in comparison, obviously. Kate holds a Bachelor of Science in Molecular Biology from the University of California and a PhD in Cancer Biology from Stanford University, from the departments of biochemistry and microbiology and immunology. And she conducted her undergraduate study on HIV-1 integration in infectious disease laboratory at the Salk Institute. She worked as a fellow and PI at the Whitehead Institute for Biomedical Research, and she headed a laboratory of 14 researchers studying viral disease that primarily affect Central and West Africa. She has some amazing stories from those experiences. She joined us. I'm not sure we did the world a favor by taking Dr. Rubins out of that world, where she was an incredibly productive scientist. But she joined us in 2009 and completed her first space flight on Expedition 48 and 49 in 2016, where again, she became the first person to sequence DNA in space. It was a very happy coincidence that that experiment flew with Dr. Rubins. She spent 115 days up there. She conducted 2 spacewalks and just performed splendidly. And Kate, I'd like you to talk a little bit about performing hands-on genomics research in space. You were the laboratory technician. You're also a scientist. And if you could help us by painting a picture of what it's like to conduct that type of research in microgravity and with the other constraints on the ISS.
Yeah, so when the sequencer guys came to me actually a few years ago and said, hey, we're thinking about flying a sequencer in space, my first thought was, I don't know why you want to do that. There's plenty of good sequencers on the ground. We can freeze samples. We can get that back down to the ground. What are the drivers? What are things that you would really need to look at in real time, and why would we do this? And there's 3 answers to that question. One is that a pretty obvious answer is the microbial and environmental surveillance. So right now, all of our microbial surveillance that we've done on spacecraft has always been culture-based. We've always put these microbes through a culture-based selection, and we know that we are looking at this much of the microbial world when we force them to grow on culture plates. So sequence-based analysis, genomic analysis of microbes gives you a much broader and richer picture of what the microbial census actually is. The culture-based methods also tie us only to times when you're undocking a vehicle and can return culture plates to the ground and are not really a real-time analysis. So a sequencing project on board could actually look at a real-time determination of a patch of green fuzz that's growing on the wall, and you could get that answer within about 48 hours. And so the experiments that we did were proving already prepped libraries and just demonstrating that we could, with a small microfluidics device that's about half the size of your cell phone and just plugs into a tablet, could we actually do, I said, where is this going to fail? Where's it going to break down? And then we'll go revise the equipment and we'll work on it. And it didn't, billion base pairs later, we said, well, let's go get 2 billion base pair sequence because it's not failing. So it actually worked incredibly well. And the subsequent experiments have even gone as far to demonstrate sample prep on board. We can use some very quick methods to prep the libraries. And then going straight from a swab material and being able to identify that organism. So there's still a lot of work to do before we replace our clinical environmental monitoring, but this is a really interesting way to look at microbial census on board. The other way that you would use this is if you have a biomarker that's important in real time that you want to analyze. And the sequencer was particularly named a biomolecule sequencer because we didn't want to say it's a DNA sequencer or it's an RNA sequencer or it's something that can determine epigenetic modifications. It can handle all kinds of different molecules. So it's a platform, essentially. And so you pick your biomarker, and we can go look at it. You can also look at things where you don't necessarily know what your biomarker is, but you're looking for a correlation to a clinical or a subclinical parameter. And you can get that data. The process to prep it, the experiment on board, is not more than an hour to set up, and the data comes back. Sequencer runs for about 48 hours, and the data comes back to the ground, and you start analyzing immediately. So I think there's a lot of opportunities for starting to look at real-time monitoring of astronaut health and potentially intervening with the countermeasure when you're actually in real time. The third reason we wanted to do this is that right now we have this great opportunity to send frozen samples back down to Earth, but we all know that soon, hopefully, we are going to be on missions that don't have that as a prerequisite. And we know that the technology at that point isn't going to be the sequencing technology we have right now. But the idea is to start right now and to get our hands around sequencing on board, start to determine what are the technical challenges, how do we do this, and already be very proficient at understanding how to sequence on board and get data back by the time that we're starting to think about putting requirements into an exploration vehicle. The other thing I want to talk a little bit about is sample processing and how we collect samples and how we think about samples, because this is very, very critical for your correct interpretation of your sequence data. So the interesting thing about sequencing or any of these high throughput studies is that you will get data no matter what. You can take anything and you will sequence it, you will get sequence data. Whether that data is good quality data or completely uninterpretable depends on a couple of things. One is how you've collected the sample. So at Stanford, we struggled a long time we were looking at peripheral blood, mononuclear cells, and gene expression. And the mixture of cells is profoundly influenced by who is in that mixture. So neutrophils have their own component of gene expression that's very strong. T cells have a different one. And so as you're taking samples and trying to compare them, you can end up with a very, very expensive CBC and differential. You're not actually looking at what each cell is doing because you're looking at a mixture of cell populations. We're starting to have computational models that can start to tease some of that apart, but those are not going to quite catch up, I think, ever to the degrees of freedom that you have in a mixed cell population. So this is very important for gene expression. It's a little less so for the DNA-based studies because those are more census studies. They just care who's there. So for microbes, right now, we just care who's there. We want to look at their genome or their 16S RNA. For mammalian cells, if we just care how many neutrophils you have, you wouldn't use sequencing to do that. If you want to see what those cells are doing, you have to get it down to a single cell type. And that in the human body, as you start to look in, you say, well, we'll get down to CD4 cells. There's a lot of subtypes there. So as we think about sample collection, you have to think about what you're putting in the tube. And at the moment you put it in the tube and freeze it, all cells lyse, the content mixes, and it's now very difficult to deconvolute. So we are probably going to want to start thinking about things like cell sorting. Single cell analysis is still, people are working on it, but it's a little bit in the future. Our capabilities right now on Earth are very exponential in terms of how many bases you can sequence. We can sequence a lot, and it's getting cheaper, and it's getting cheaper at a rate far more exponential than Moore's law. So that's where we are right now. We're not quite as good at working at that real minimal sample input. So when you're dealing with sub-nanogram sample inputs, that technology is catching up and will be here soon. At that point, you can then start looking at single cell analysis and what are each one of these cells doing. But we want to be extremely careful when we design experiments that we don't just draw blood and look at the omics of gene expression in astronauts, because we'll never be able to sort that back out again. We also really need to think about the metadata that accompanies each of these samples. So for example, Scott Smith's food intake application. If you go do just a gene expression profile in blood, one of the most important things is how long has it been since somebody has taken a meal and where they are in their circadian rhythm. So understanding for the astronaut when they've eaten, when they've exercised, what the temperature was that day, how much CO2 was on the vehicle, all of those kinds of metadata go into interpreting what went into this gene expression. And we also need to think about what we're going to do with space station, not just as the current set of research that's working incredibly hard to buy down risk, but how are we going to start to bank samples so that we have samples in the future, when the phenotypes start to catch up and we actually can link more phenotypes with genotypes, that we have a set of samples that we can go back and retroactively say, hey, we're going to pull 10 astronauts, this cell type, and go run this profile and correlate it to some phenotypic outcome. In order to do that, we need to proactively collect and bank samples. And so we want to be thinking about the sample type, sample accessibility, what kinds of cells make up your sample, and then the other part that we do right now on board in a pretty disjointed fashion is the temporal sampling. So we are crew time limited. We have resource limitations. We say, hey, your study's really lucky if you can get 3 or 4 sample points. And I would suggest we may want to start rethinking that for some of these big sort of banking efforts, that tasks on board are, once you start doing them and they're repeatable, get much easier. So if a task is performed 3 times, you open the procedure and it's sort of, it's going to take you quite a bit of time each time. When it starts to be something that you know how to do every week, you could imagine waking up every Monday morning and taking a saliva sample, a urine sample, a blood sample, a stool sample, and a skin swab. And you just know every morning you're going to do that, and after about 3 or 4 weeks of this, you're going to get very quick. This is not going to be a crew time hassle. You also then have to think about where all those samples going. Well, we store our samples in giant tubes right now. We don't use 96 well plates. We don't use 384 well plates. We can start using extremely high density methods to store this massive increase in our time course sampling. So I think the keys to where we're going on station is thinking about exactly what it is that's going into your sample, how we're going to collect it, and how we're going to increase. We've increased by a few orders of magnitude the number of genes that we can look at from this single gene experiment to 50,000 or so. Now we want to increase our temporal sampling. We want to make sure we have the right metadata associated with it, and we want to look at what the cellular constituents of our sampling are.
Okay, thanks very much. I think the overwhelming message I'm hearing is that the payback is not near term, but the urgency for collection is, and that in itself is going to be a very complex planning process. Dr. Virginia Wotring, she is Associate Professor at the Center for Space Medicine in the Department of Pharmacology at the Baylor College of Medicine. She's also the Chief Scientist and Deputy Director of the Translational Research Institute for Space Health. Dr. Wotring earned a bachelor's in chemistry and biochemistry from Florida State University and earned her PhD in pharmacological and physiologic studies from St. Louis University. Her graduate and postgraduate research centered on structure, function, and pharmacology of ligand-gated ion channels of the nervous system. I bet that's really important. Her current research involves examining the changes in physiology and pharmacology that occurred in the confined, closed microgravity and elevated radiation environment of a space mission. Other projects include studying women's health treatments during spaceflight missions and examination of genes and proteins altered when organisms live in a spaceflight environment. Her book, Space Pharmacology, was published in 2012 by Springer Fairlog as part of their Briefs in Space Development series. And Ginger, you're kind of uniquely qualified to talk about the role of genomics in precision medicine, in particular for astronauts, having an idea of what happens to biochemical response to weightlessness and how that might map to pharmacological actions for the drugs that we most likely prescribe. So I'd like you to talk a little bit about that.
Thank you so much for giving me the opportunity, Mike and Mr. Abbey. Yeah, so we haven't mentioned yet, there's a special branch of genomics, and that's pharmacogenomics. Drug companies realized when we figured out that there were different genetic variants of molecular targets of drugs and the drug transporters that move drug from one place to another in our bodies, and most especially the enzymes that metabolize drugs, there are differences between people that can be explained by their genetics. And if we know what kind of person we're treating, we can use that knowledge to direct drug choice and drug dosing. And major drug companies have been working on this for some time. It became a big push around 2010. And at this point, we're seeing dozens of drugs and genetic test pairs that are on the marketplace. Most of them are made for drugs to treat cancer. This has been developed and driven by drug companies. They're looking to treat people who need treatment. They're looking to make money for their companies. So they're starting where they need to for their own company. And at this point, they have developed tests for SNPs, single nucleotide polymorphisms mostly. And these are what the Smith 1-carbon study was looking for. Not an entire genomic analysis of a person, but just a very short stretch that we know it's already been identified to be important in an action of a drug. So you can run this little test, and the prescribing physician can get information about whether a particular drug will work for you. It may be that you need to get a different drug. It may be that we can choose a drug that's ideally suited to you. It may be that we can use this information, in some cases, to dial in a dose rather than having a patient go home with a prescription and the doctor says, try it, see how it works, get back to me afterwards. We'll talk about how well it worked and if you had side effects. We don't have to go through that as often anymore if we can get this information about a patient and dial in the dose. And just to give you an idea of perspective, we have at least one drug now, it's an anticoagulant, where there's a 10-fold range in the recommended dose for different people. 10-fold range. So for some people, you could give them actually a poisonous dose going by averages. And that person would suffer tremendous side effects and be very uncomfortable. While for another person, they might get a terrible underdose if you go by the averages, and they might not get the efficacy that they need. So this test can be used to dial in the right dose and eliminate some of the trial and error that physicians have been forced to use in the past. So the state of the art here, we've got a few dozen cancer drug genetic test pairs that are on the market now and being used every day. There are a couple of anticoagulant drugs that we also have tests for. There's a panel that is quickly becoming popular. It looks at a series of 6 cytochrome P450 genes, and these have been linked to the efficacy and side effect and dosing descriptions for a handful of different behavioral drugs, mostly anti-anxiety and antidepressants. This is really becoming popular. These panels cost a few $100s to run, and results get back to the doctor in just a few days. It's a simple SNP test that can be run by qPCR, so it's not a big sequencing project. And because it's not a big sequencing project, it gets around some of those privacy and personal health issues that we were talking about earlier. This is really focused information. For the spaceflight world, I'm thinking, and I've been talking to Dr. Bayous at the JSC Pharmacy about this on a pretty regular basis. We've been watching this field develop over the past few years. Right now, the tests that are available, the pharmacogenetic tests, are not focusing on any drugs that are really used by the astronauts. But I think the day is coming, and I think the day won't be very long down the line. This has been developing since about 2010. We've made a lot of progress in this amount of time. And because of the recent opioid crisis in the news, I have a feeling that pain relief is going to be the next target for these drugs. They're identifying the right kind of pain reliever for an individual, putting them at a good dose, helping treat people's pain and maybe keeping them off the more addictive pain relievers. I have a feeling that there's going to be a huge market push in this direction soon. And there we're starting to get into some medications that astronauts do use. And then I think it's just a matter of time before we work further down the line into drugs that not only crew members use, but more healthy adults on Earth use every day too.
Okay, thanks very much. That sounds like something directly applicable to the larger commercial tourist trade that may be coming in the next several years. So
absolutely.
I think
if we could give people a motion sickness drug
right.
that doesn't make them sleepy, how wonderful would that be?
Yeah. Okay, very good. Thank you. All All right, last, absolutely not least, Dr. Smith Johnston, mostly known to everybody, but he completed residencies in internal medicine and aerospace medicine from Wright State University and a Master of Science in the Aerospace and Preventive Medicine field. Dr. Johnston is a member of the clinical faculty at the University of Texas Medical Branch, and he has spent most of his career as a medical officer and flight surgeon for the NASA Medical Operations Branch at the NASA Johnson Space Center. Over the last 25 years or so, he has supported medical care of the astronaut corps, their families, and retired astronauts. I think it's safe to say Smith is known as the flight surgeon's flight surgeon. He has worked absolute miracles in returning people to flight status in a very safe and effective manner, and he has worked over 25 space shuttle missions and worked long-duration missions as well. He served for several years as the medical director of the NASA JSC Aerospace and Occupational Medicine Clinic. He is presently the lead of our astronaut medical selection and retention standards group, and also fatigue management and human health performance and longevity programs. Smith is the go-to guy for medical standards. He has worked both within JSC and internationally to help coordinate medical standards for long and short duration flight. And if any medical problem has come up, Smith has probably worked it and tried to figure out how it could be flown safely in space or not. And I've asked Smith to talk about the use of genomics in long-term health assessments and risk prediction and strategy. And if you would, please touch on the very sensitive topic of using genomics for selection standards.
All right. Well, first of all, thanks, Mike. Thanks for that introduction, and thanks for this panel. It's an honor to be here, and it's an honor to have been asked to participate. Like you said, I've spent about 27 years at JSC and 23 years at NASA, and it's my other honor is I get to take care of one of the best patient populations in the world. And it's also just such an exciting time in medicine. I mean, when I was training in med school in the 70s, we just got our first CAT scan at Emory University, and that was, whoa, and now we're sequencing genes up in space. So it's just an incredible time. As far as the standards go, I've worked with our tremendous international partners to develop standards for selection into the astronaut corps for astronauts, and for up to a 15 or 20-year career, and then with annual certification. And it all comes down basically to risk mitigation. Our first priority is to the mission, and we've talked about this. Our immediate mission at hand, which is to have the astronauts and cosmonauts as healthy as possible and performing at their best. But I would say also that our overarching mission is also to take care of our retired astronauts. Those are, and discuss longevity, because that's how we're going to figure out whether or not this space environment on a long-term basis has been deleterious to our astronauts and cosmonauts. We have 14 astronauts in their 80s right now, and so I'm going to end with some notes on that, because I think that's something we really need to focus on. Genomics, genetic testing, I'm all for it. We're all for it. We need to do it, and as much as possible, and of course in a way that these folks can tell us the best way to do it. We need to know what our strengths and weaknesses are. I mean, why do you want to know if you have an ApoE4 gene for Alzheimer's? Well, because on an individual precision medicine basis, you can take that. Instead of having a 7% chance of Alzheimer's, you now have a 32 to 40% chance of coming down with Alzheimer's. But you, with therapies and fitness and diet, exercise and medications, there's now data. You are starting to see the slowing and possibly even the regression of certain Alzheimer's, or at least the endothelial wall function of your arteries supplying your brain that are helping you to transport those tau and amyloid plaques out of your brain. And the reasons we sort of, and genetic testing is something that it's not new. We've been doing it from day one, and it's called one of the most important things we do clinically when we see a patient is get a history and get a family history. You know, if I've got somebody who has a family history of somebody with colon cancer, or breast cancer, or coronary disease at an early age, then I'm going to go, whoa, I don't really need a genetic test, but I sure would like a genetic test to get the genetics and then be able to follow and see what the phenotype ends up being. And that's the exciting part about the fact that now, when we started sequencing the first person, total genome sequence was, I think it was 2007. It was Watson of Watson and Crick. It took us 2 months and $1 million and $3 billion worth of research to get there. And now it's about $1,000 in 2 or 3 hours. You can do an entire gene sequence. So, and 23andMe is $200, and you can go find out a few fun little genetic markers that you may have that you might not want to pass on to your kids and other things. So it's out there. We have several astronauts who are adopted. We have no clue what their family histories are. And we also have astronauts who are showing up going, I went out and got genetic testing, and here's my genetic testing. And so the NASA policy, they've been working on that for several years, and I believe we've signed the policy. We've not implemented it, but we think we have a policy document that we're pretty close to, and that's very exciting. So I'm all for it. I'm also all for getting our retired astronauts back to the clinic. Bonnie mentioned yesterday that we have the ability to come back to the clinic, and the last few years I've been at NASA, we've been trying to focus on a really value-added physical or evaluation at the clinic, or making it as easy as possible. Say you don't want to come to the clinic, but you just got a battery of tests done at your private physician at Johns Hopkins or Mayo Clinic or the Boise Clinic. You're able to give us a call, and we will get those documentations sent to the clinic, and that information will be entered into your EMR. Some people come and just get the part 1 of the physical, which is the laboratory evaluation, and the blood work, and the other evaluations we do. We have an in-house ultrasound machine that an ultrasound machine in the hands of Shot Sarkisian is as good as a full-body CAT scan in certain terms. We've really pushed the envelope on our use of ultrasound. We do carotid intimal thickness to follow carotid artery and atherosclerotic changes. We have every Wednesday, we have a clinic that any retired astronaut can walk into, a musculoskeletal clinic, where Rick Shearing is actually teaching an orthopedic fellow at this astronaut gym, seeing our astronauts to use ultrasound to help diagnose and manage and treat musculoskeletal injuries. We live right next to the world's largest and one of the best medical centers in the world. And so we're able to utilize a lot of the research and the clinical programs that MD Anderson, Methodist, and other use. If we have a program that we've instituted based on their data, it's not standard of practice. It's not being paid for by insurance companies because it's not standard of practice. But we monitor CA-125s for ovarian cancer. We monitor them on a yearly basis, and we're hoping if we see a spike, just if you saw a spike in your prostate-specific antigen, you retest it, and if it's going up, you might be in that window of opportunity where you could prevent an ovarian cancer, which has deadly consequences. So I'd like us to try to get the word out that we are trying to have a value-added clinical exam for our retired astronauts as well as our astronauts, and sit down and get that word out. And finally, I'd like to talk about, it's important to know the genotype. I mean, genes are 20 to 30% of us. The rest of it, the environment, what we live, how we live, what we eat, breathe, fitness, strength, psychological well-being, that's the 70 to 80% of us. And so knowing that 20 to 30% and then watching how we handle what happens in that 70 to 80% of our environment affords us that risk mitigation knowledge we know on as far as how to intervene with certain medications, certain therapies, such as fitness, certain diet, and that sort of thing. But finally, to really know what the phenotype is, to know the phenotype, I want to come back to what's been brought out before. We need to have autopsies. We need to have some sort of autopsy program, and we've tried to do that in several instances, and I think, Mike, we talked yesterday, we're trying to really formalize that because we don't really know. If someone dies of coronary disease, we don't know if they had a cancer in some other organ system. So it's really important that we institute this, and then finally, the other thing about DNA sequencing is you also get DNA methylation, and that's kind of like the hemoglobin A1C of aging. It gives you a 3-month period of what your DNA damage was, and that's a good way to look at aging. We also have worked with Susan Bailey out at Colorado State, and we've developed a test for about $126 that does telomere length. And you can go online and get your telomeres done, but this is probably, as high quality as you can do. And telomeres length is a great indication of how well you've aged over the last year. So that's another area that I'd like to try to get involved, and we have some people who've done their telomere tests. And then finally, one of the things we owe all of us, and the lay public and the lay medical organizations out there, is we are learning incredible things by taking care of these individuals and by following these individuals throughout their lives. And this word needs to get out. It needs to be translated that you can reverse coronary disease. It can be done. And we've known that since 2011, since the studies in New England Journal came out. They've actually known it a little bit before that using intravascular coronary ultrasound. But we're so bombarded with information. There was a IBM Watson study done that showed about 45% of clinical medicine only 45% is evidence-based to date. And it takes 17 years when an evidence-based practice is discovered to become practice. And we need about 29 hours a day to keep up with our professional insights, to be on top of things. So we can't do that. So that's part of our job is to take what we learn from our astronauts, incorporate the use of, I just look at genomics and gene testing as just another incredible tool for us to use. And I don't see any difference in it and not being able to firewall it and use it clinically as anything we do, such as calcium score testing for cardiac disease, finding early asymptomatic cardiac disease. And that informs us that, yeah, you might want to get your total cholesterol below here and your HDL here and your C-reactive protein inflammatory markers here, and you might want to change your diet, and we might be able to sequence your genome and see how we could, facilitate that. So that's my tirade on how I want this to happen, and thank you for letting me speak.
All right, well tiraded. Okay, so let's go immediately then to. I have a raft of questions, but let's go to the attendees and. Box.
Oh, I got the first question. That's great.
Well done.
So, Smith and Jen and Craig, and you talked about the astronaut data being, sequencing for selection. I thought you were dodging it, but I think your answer was yes. If we keep it private, you're going to use it for selection, and I think you should. I actually have always thought that, at least I personally think, all of our data, health data, should be, it's something you signed up for. And so if you're going to go to the moon or Mars, it becomes even more important. All your health history, your whole complete genomic sequence. I just wonder when and if we can get to a point where it's an open book. That's what you sign up for. Then you can understand your health problems, and thank you for working on mine all these years and keeping me alive and keeping me flying, because you guys do that. But it just seems to me that there's a range from those who don't want to totally protect privacy, and Jen, you talked about the families, and that's an important point, but I'm just wondering when we're going to get to the point where that's just, hey, you want to be an astronaut, you want to go to Mars, you're an open book. When will we get there, or should we?
I didn't answer that question. You asked me to speak on that.
Yeah, just for astronaut selection,
good,
right.
Good. Yeah.
But when, what range should we be an open book when we do this job?
I think until the laws and the fact that you can lose your life insurance. I mean, you can lose, certain laws have to be changed. So I think, yes, I think we can firewall it. Yes, I think the policy is saying we're going to do genetic testing, but we're going to do it for surveillance. Okay. And we do that already. Like I said, if you came to me and said, I had a brother who died of early coronary disease, but your labs look great, your treadmill's fine, you have 0 calcium on your calcium score, then we're just going to probably treat you a little more aggressively on your cholesterol levels and your fitness levels and get your C-reactive protein low. And if we had genetic markers to help inform us of that, like you do or don't have a lipo then we might use a different kind of, which is a carrier protein, lipoprotein, that if you have it, that's not good. But we can treat that with medicines. We would just target your LDL much lower. And there's some new drugs. I'm going up to Cleveland Clinic this weekend to sit down with their chief of cardiology. They're in a trial right now with lipo lowering medications, and they have the PCSK9 inhibitors that lower lipo So we have a few folks in the Corps and in management that we might be using those on, or even myself. So to answer your question, I think to start the program, I think we use genetic testing, but we use it for surveillance. And we don't use it for selection. Now, down the road, when we get smarter about it, sure, there might be someone that, and we do that with radiation in a way. We say, I'm sorry, the risks are too high for you to do a 1-year mission. We've done that. But we're not there yet with our genetic testing, I don't believe. But that down the road, we're not to Gattaca, and hopefully, I don't know if we'll get to Gattaca, the movie, but so I would say right now it's for surveillance. Like, we would screen the core, we would then bring the core in, and then for their first annual, we would get gene sequencing. And then we'd use that information to inform them and their families, hey, we found this, we need to follow this, we're not sure what it means, or hey, this is what you've got, you probably need to do this, this, and this. But it's not going to impact your career, hopefully. Hopefully, he says.
And just to add on a little to that, Greg, I think we NASA will not be on the leading edge of these applications. I think, like for the reasons Ginger mentioned, there's broad societal benefit. It's a very emergent field. And from a NASA perspective, we're kind of looking at risk and benefit. And the benefits are just going to keep going up and up and up. And it'll become more socially acceptable. And I think, as Smith says, laws will change, that will build in protections. And so I think in time scale, I'm just making this up, 5 to 10 years. I mean, it really depends on how quickly a lot of this is assimilated into practice now. But I think that's a kind of a time scale where we might be much more involved in the applications. Go ahead.
Yeah, thank you. And you hit on a lot of really interesting issues. And one of the things that comes to mind as I listen to it is sometimes with the testing that we do currently, we're looking for an end state to intervene. But sometimes when we're looking at the genetic expressions that are occurring, those are adaptations that are occurring, some of which may look bad at the early stage because of the way the body is attempting to respond, but it's bringing you to a point that would be better. So depending on when you tested and sequenced it, it may look bad, and you may then decide to intervene. Had you waited, you would have come about to a better result. And a great study, well controlled, that looked at just exercise. Exercise 6 months, 7,663 different genetic upregulations and downregulations. So if you started and looked at, well, what's the person look like today, or midway through that exercise, they would look different than they did at the end of that 6-month cycle, where they were now more fit. A lot of the underlying physiological biomarkers that we would care about would now be better aligned. That person would show a more fit profile. So that's part of the challenge, I think, because it's easy to kind of become stochastic in it's like either good or bad. test. We're going to get it, we're going to get a test, and it's going to show us. But there is an adaptive process for the reasons that were cited, the methylation and the epigenetic expression. So that's part of the challenge as we do this, is to maybe come back with a strategic approach that says, what are the processes that need to be looked at, and how will we know when we're in the right path to the right end state? Because as we sample, we're going to get answers. But the answer may be an early answer that isn't the best answer. So I just offer that.
And so when we try to discuss the complexity of the time course of change, given the plasticity in the human, and that so few genes appear to be absolutely deterministic, right, that you're really trying to understand the different interactions, to your point of what is acute phase, what is chronic phase, even at the phenotypic level, going back to suppressing all inflammation is not good, right? These are signals that need to be there to cause repair and end up in a state that's more appropriate for what your body is managing, whether it be exercise or even cardiovascularly. So I think the points that Smith was making are interesting to me, and even Ginger, because from a terrestrial perspective, the end states of particularly cancer have gotten a lot of attention, and the intense mapping, once those people are identified, and then that surveillance goes up and managing their treatment and outcomes. Same with cardiovascular. So we're working on the healthy spectrum, which is where you're seeing a lot of that flux of your body trying to manage these complex stressors. And some of these things that we're identifying potentially even in spaceflight aren't unique to spaceflight. They're a stress response. So we look for analogs where we can dig in a little bit more deeply, control some of the other variables. But there may be some that over time we will discover are spaceflight unique for the unique stressors of spaceflight. But then that time course of saying, again, yesterday it came up about first do no harm. You may need to watch this thing and be uncomfortable watching it do its thing, because the body is amazingly resilient. And if the genetics prescribe such, again, a deterministic outcome and had that kind of rigidity, we would be dying a lot more, right? But we're not, because we even go back to the point of, okay, let's step back. We have 500 and some odd people who have survived spaceflight and flourished afterward, So when you just look at that level, we've got some data there epidemiologically that you say not knowing the noise and being uncomfortable with not knowing the mechanism is something the scientists and the medical people and the smart people in the space program, including the astronauts, struggle with, which is I can't ignore the fact that we have successfully done this. The human body has successfully achieved it. We want to make it better or more certain. I think the early identification of processes that look like they're struggling to accommodate and then suddenly don't. I think the tertile, was it you, Dr. Dingus? Yeah, that came up. When you have populations that segment that way, you really would like to have that insight to help the people who are in the lowest tertile and not able them to fly, because they might have some other characteristics that they would benefit from and the mission would benefit from. But yeah, separating that out. And the robust kind of research to do that is really in partnership with, as you know, Tom, the DOD, the academic medicine, and saying NIH, NCATS, like where do we have these partnerships where they're getting much larger quantities of data, and then we can go in and be more strategic about what we need to do in spaceflight because no one else is having that perturbation. Now, Greg, you asked about laws. There is no doubt, like the laws are in place because we're globally not on the same page with what this data means and how to use it. And when it comes to insurability for you, your family members for the rest of their life, it's a struggle. So the GINA law is there to protect you. And really, it's not functional. We're all inherently flawed. So you go looking hard enough, you find a magnifying glass that can go measure it, you're going to have an indication that something is not beneficial. But to hold that against someone without really understanding that ultimately will mean a bad outcome. So we're not in a place where we can allow that, because we're definitely not on the same page. And the last thing we want to do is inadvertently set off a chain of events for people who are going to live in the time period before that's all sorted out. I'm often like, well, if you genotyped everybody and just threw it all out there, insurance would be so overwhelmed with the flawed data, no one would be insurable. So, okay, then we reset the baseline. But that's not the position we're in right now. So we're grappling with it for sure.
Actually, we got time for just two kind of shortish answer ones. Guillaume has been waiting patiently, and David will come. We'll finish up with you.
Yeah, I wanted to comment on the Rubins, on the comment by Dr. Vautrin on the testing of individual response to pharmaceutical. That's something that we have started already 1 year ago for the ESA astronauts to test them on the medication which are in the ISS medical kit. We started 1 year ago, and that's because it's linked also to the annual examination. That's something we will have the final results pretty soon, and that's something that we intend also to study, to open.
So quickly, is that required, or is that science?
That's an ESA internal study on the medical as a
Is it required, or is it an investigation?
It's a medical investigation, which means it's not
Is it required? Or, okay.
No, it's not mandatory, which means that we have all astronauts who have accepted to do that. So we have we have an N equals 7, and we will have the end results here by the end of the year. So probably that's something that we will be able to discuss pretty soon. And we have seen already interesting variation on the response to the ISS medical medications. On the other side, I wanted also to comment on what Smith said about the importance of genetics in the medical evaluation. And pretty much the European position is exactly along that line. There is one point for which we might not yet at the point that that Smith mentioned, which is to include the complete sequencing after 1 year after selection. But that's certainly a point I would welcome to have as a discussion point item for the medical standard working group. Because I think that's also something. Currently, we don't have yet all the information to see how determinant is an information and how critical and how to exactly handle that. So I would have a certain cautious position up to now. But I think to have that information on hand, so to be able to extract valuable information when we can, is surely something that we need to think of.
Okay, great.
Yeah.
Dr. Dinges, bring us home.
The great panel, this is an exciting time, and that's really my point. Well, we're running into all sorts of extraordinary things now that are difficult to understand. So it's true that we focused on genes, but I can put scientists in front of you who would say, gene schmeans, the proteomics are where it's at. And that's where you need to be looking. And then there's people like me who would say, no, you need to look at metabolomics. We found a hit there in rodents and humans, and it showed a state. It tracked the perfect state of sleep debt and recovery. And it was a small N, but it got into Proceedings of the National Academy of Sciences because it was unbelievably reliable on 2 continents in animals and humans. So we're in this really exciting time, and I think the watchword here is surveil the literature. Pay attention to what's going on, because there's an enormous amount out there. It's a little overwhelming, as some of the commentators said, but I think it's worth watching and looking for opportunity here for NASA. So you don't have to find it all, but you could capitalize on the discovery somewhere else. And you're not the only agency. Now, you must have set this off or something, Mike, because now other agencies that I thought were transportation agencies and that all want to know about omics markers for, could they stop you on the highway and check if you have an omics marker for alcohol or an omics marker for sleep loss? Or so we're actually starting to see what might be ridiculous, but nevertheless serious efforts to look at omics in another way. I think it's very exciting, and I think looking at omics broadly and continuing to surveil literature as well as do the work NASA is doing is just spot on. So good luck.
Well, thanks very much for the good comments. I'd like to think we started all this trend, but I think there was a lot more attention to how much Neanderthal DNA is in Ozzy Osbourne, because that got the attention of the lay public, and I think that's raised the awareness.
How much Neanderthal do you have in your spine?
That's classified. That's definitely need to know.
I've got
All right, so I'd like to wrap up here to be sure we have a reasonable break time and get the next panel going on side. But you know who knows what now, so feel free to take advantage of them all, volunteer their availability for the break. All right, thank you very much.